I'm going to go over part C of chapter 3, cells. The first thing we're going to talk about is what is a cell and what is a cell made out of. So the first thing that a cell is made out of is, we've talked about the plasma membrane, but inside that plasma membrane is the cytoplasm, and it's located between the plasma membrane and the nucleus. It is called cytosol, and it's water with solutes in it, so protein, salts, and sugars, as well as organelles. And this is the metabolic machinery of the cell. We need to remember that metabolism is the balancing out of homeostasis. They also have inclusions, which are granules of glycogen or pigment producing cells, as well as lipid deposits, vacuoles, and crystals are all found in that cytoplasm. So cytoplasmic organelles are going to be either membrane-bound or non-membrane-bound. So membranous ones are the mitochondria, peroxisomes, lysosomes, endoplasmic reticulum, and the Golgi apparatus, while non-membrane-bound ones are going to be cytoskeletons, centrioles, and ribosomes. The mitochondria is a double membrane structure that has these shelf-like cysteines, and these literally look like zigzag bookshelves within the mitochondria. This provides most of the cell's ATP via aerobic cellular respiration. So they are going to use phosphate to phosphorylate or adiphosphate to ADP or adenine diphosphate to make it adenosine triphosphate. And they also contain their own DNA and RNA, and they reproduce or replicate independently of the cell. So we can see here the mitochondria. You can see those shelf-like appendices, inner workings. So we've got the outer membrane and inner membrane, which are the cysteines, as well as its own matrix, and it has its own enzymes as well, and it makes its own ribosomes. Ribosomic enzymes, it has its own RNA and its own DNA, so it makes its own inside. Ribosomes are granules containing proteins and rRNA. They are also the site of protein synthesis, so we're going to see them take mRNA and synthesize it into an amino acid structure and then folding it and making it a protein. We also have free ribosome synthesis soluble proteins. So these just float around in the cytosol. Then we also have membrane-bound ribosomes. And these are found attached mostly to the rough ER. These synthesize proteins that cannot be incorporated into the membrane or exported from the cell. Next thing we're going to talk about is the endoplasmic reticulum. This is an interconnected tubes and parallel membranes that enclosing cysterines. They are continuous to the nuclear membrane, and there are two varieties. We have the rough ER and the smooth ER. The rough ER is going to have a bunch of ribosomes dotted around it, so it's going to look like a rough texture, and you can remember rough with RNA and ribosomes. So you can see the smooth ER is nice and smooth, while the rough ER is going to have those ribosome textures on the outside. So the rough ER is great for synthesizing proteins, while the smooth ER is great for synthesizing steroids and hormones. So the external surface is studded with ribosomes, manufactures and secretes all proteins, as well as synthesizes integral proteins and phospholipids. So this is really important for the continuation and building of the plasma membrane, as well as keeping that integrity strong. Smooth ER is tubules arranged in a looping network. It has enzymes that produces enzymes for specific functions, and you can see this in the liver. It has lipid and cholesterol metabolisms. It breaks down glycogen, and in the kidneys we can see the detoxification of drugs, pesticides, and carcinogens. They also synthesize steroid-based hormones, and in interstitial cells, they help with absorption, synthesis, and transports of fat. So these are going to be things like our stomachs. Our stomachs, we only have one. Our small intestine, our large intestine, and the duodenum. In the skeletal and cardiac muscle, it's going to store and release calcium to help produce and maintain an electrochemical gradient, as well as keeping that muscle moving. The Golgi apparatus is a bunch of stacked and flattened membranous sacs. They modify, concentrate, and package proteins and lipids. They also have transport vesicles that they take transport vesicles from the ER and fuse with convex cis face of the Golgi apparatus. So they're going to take proteins from the rough ER and they're going to package and modify those proteins and lipids so that they are functional. Proteins that pass through the Golgi apparatus then turn to transphase, which is just moving the molecules around, and then secretory vessels leave transphase of the Golgi apparatus stack and move to the designated part of the cell that goes outwards. So they go through it, they go on one side, and they go through it to the other side. One side's called the cis phase, and the other side is called the transphase. So you can see here the protein containing vesicle pinches off of the ER and migrates to fuse with the membrane of the Golgi apparatus. It then fuses with the Golgi apparatus and is modified and then is packaged into a different vesicle depending on the ultimate destination, and is either secreted out of the cell or is used within the cell. So exocytosis incorporated into the plasma membrane or can be used within the cell. So lysosomes are spherical membranous bags containing digestive enzymes, and this is typically an acid hydrolysis. They digest ingested bacteria, viruses, and toxins to remove them from the cell. They also degrade non-functional organelles that have died and have been replaced. We can also see them participate in the breakdown and release of glycagon, as well as the breakdown and release of calcium in bones. Destroyed cells from injury or non-useful tissues are going to be broken down by lysosomes in a process called autolysis. Our endomembrane system, the overall function of this is to produce, store, and export biological molecules, degrade potentially harmful substance. So all of the organelles that we have talked about are endomembrane systems. So we see the smooth ER, the nucleus, the nuclear envelope, vesicles, plasma membranes, lysosomes, rough ER, Golgi apparatus, and the transport vesicles. Those are all part of that endomembranous system. Peroxisomes are membranous sacs containing powerful oxidative and catalyst enzymes. They detoxify harmful or toxic substances, and they neutralize dangerous free radicals that are these free radicals are highly reactive chemicals with unpaired electrons that can go in and damage DNA to make it unrepairable and even cancerous. Then we have our cytoskeleton, which is an elaborate series of rods throughout the cytosol that help keep everything in place. So we have microtubules, microfilaments, and intermediate filaments are the three different parts of the cytoskeleton. Depending on what they're used for is which type it will be. So microfilaments are dynamic actin strands attached to the cytoplasmic side of the plasma membrane. These are mostly involved in cell movement as well as changing shape. They also assist in endocytosis and exocytosis. Once again, that's still changing shape of that plasma membrane. So we can see it here. It is literally just little actin subunits all together. They're about 7 nanometers long, and they form a spherical, they are made of a spherical protein subunit called actin. You can see them here in blue. They surround the pink nucleus in this photo. So you can see all those blue little hair-like strands in that filament. Intermediate filament are tough, insoluble, rod-like protein fibers. They resist pulling forces on the cell and attach to dimnosomes. So we can see them here. They're the purple in this photo. They're about 10 nanometers long, so they are larger than microfilaments. They are insoluble protein fibers. They almost look like a branded rope. They have fibrous subunits. Once again, you can think of it, fibrin is like a fiber in a shirt. And then microtubules are hollow tubes. Most radiate from centrosomes, and they determine the overall shape of the cell and the distribution of organelles. So this is what's going to hold all of the organelles together and maintain most of that shape of that cell as well. So you can see them here in yellow. They're hollow tubes of a spherical protein, and they're called tubulins. So you can remember microtubules have tubulin in it. They appear gold, and they have that network surrounding the cell's pink nucleus in this photo. We can do all of this with fluorescent staining, taking antibody fluorescents and attaching them to these protein structures. So we also have motor molecules, and these are complex proteins that function in the mobility or movement of organelles and contraction of the cell, and these are powered by ATP. So we can see them here. We've got a microtubule of the cytoskeleton attached. A vesicle attaches to it and moves it along. They're almost like little feet. So motor molecules attach to the receptor, and the vesicle or the organelle practically walk across the microtubule of the cytoskeleton. And they also move against each other, literally walking against each other. This is how cilia move on your intestinal lining. So centrosome, you can remember this because it's the center of the cell. It's near the nucleus, and it generates all microtubules and organizes mitotic spindle division. So we can see this. It contains centrioles, a small tube formed by microtubules. So you can see it here. They're almost star-shaped. You can see the centrosome matrix and the centrioles and the microtubules coming to go attach to it to be able to divide and move how they need to. So we do have cellular extensions. So these are on the surface of the cell, and not all cells have them, but some do. So we have cilia and flagella. Flagellum is a whip-like movement that propels the whole cell. It's kind of like the tail. The only cell that has it in the human body is the sperm. While cilia are these little finger-like protrusions on the outside of the cell. They contain microtubules and motor molecules, and they move substances across the surface membrane. So they're almost like grabbing the substance and bringing it into the cell to be used. So we can see them here. We've got microtubules on the outside, connecting to the cilia and being able to move it in. We can see the longitudinal sections of the cilium, and you can see those microtubules running the length of those structures. So you see them here powered. They have a stroke method. Go grab it back in. We see these a lot in our intestinal tracts. They do grab onto things and pull them into the body. Microvilli are another smaller form of cilia. They're finger-like extensions of that plasma membrane again. They are specifically used to increase surface area for absorption, and the core of them are actin filaments, and they use these to stiffen straight up to provide that increase in surface area. So they're almost like hair. They stick up, hold them out, and you can see if a surface area of just a circle is not that strong or not that much, but if we add a bunch of indents and grooves, that almost increases. Once again, we see these a lot on things like our intestines. So the nucleus is the genetic library with blueprints for nearly all cellular proteins. Each one of your cells has a nucleus except for a handful. They have all of the same amount of DNA, and only certain parts of that DNA is expressed depending on what cell it is and what proteins they need. So it responds to a signal, and that dictates what kind and the amounts of proteins that need to be synthesized. So your eye cell is not going to produce the same thing as your liver cell does. Your eyes don't need liver enzymes. Your eyes need bactericidal fluids to help keep the bacteria out. Most of your cells are going to be uninuclear, so they're going to have one nucleus, while red blood cells aren't going to have a nucleus at all. Skeletal muscles, bone, distraction cells, and some liver cells, as well as immune cells, are going to be multinucleated, so they're going to have many nucleuses in one cell. Other things that are anucleated are going to be things like platelets. That means they don't have a nucleus at all. Red blood cells, platelets, aren't going to have a nucleus. So the nucleus you can see here, you've got the nucleus, the chromatids condense the nucleolus, and then the nuclear envelope. We also have the nuclear pores that let RNA and DNA out if needed. The nuclear envelope is a double membrane system that contains pores. It connects to the rough ER and is almost continuous, and it also has ribosomes on the outside. The inner lining is the nuclear lemma, and it maintains the shape of the nucleus. The pore complex regulates transports of large molecules in and out of the nucleus, depending on what's needed at the time. So this is going to move proteins in and out for things like DNA replication, RNA, so on and so forth. So you can see here an electron image of that nucleus. You can see those pores where things can move in. They are ring-shaped by protein particles to let things move in and out. And then you've got that next net-like structure that is on the inner surface of the nucleus that maintains its shape. The nucleoli is typically a dark, stainy, and spherical body within the nucleus. This is mostly involved in RNA synthesis and ribosome subunit assembly. So we're going to see them make that RNA as well as assemble those ribosome subunits. The chromatids are thread-like strands of DNA, 30% of it. We see histones proteins being about 60% of that chromatin and RNA being about 10%. They're arranged in fundamental units called nucleosomes, and they're condensed into bar-like bodies called chromosomes when the cell divides. So most of the time they're nucleosomes, and then when the cell divides they become chromosomes. So we can see the DNA double helix here, and it wraps around the histones, which are the beads and the strings. So it starts wrapping around them. And the nucleus, the DNA then is winding in on itself to these tight helical fibers, and then it's looping in on itself to create that chromatid structure that we see here during metaphase, the midpoint of division. And that's what we talk about when we talk about chromosomes, is that structure there. All right, that is it for today. I will see you next